Hi, everyone. I'm Shannon Wyman, the Scientific Communications Manager at Keystone Symposia, and I'll be hosting this Meet the Editors Roundtable today. I have with me editors from Cell Press, EMBO, JEM, Nature, the Company of Biologists, PLOS, and Frontiers Journals to discuss the state of the field, exciting new advances in single-cell analysis, tools, and applications, as well as tips on how to publish in these top-tier journals. Before we get started today, I'll go through some instructions on how we'll be running the session. First, please keep your camera and microphone off during this first portion of the event. We'll start with a 30-minute discussion with our panel and then open up for audience questions. You may submit your questions at any time into the Zoom chat. During the audience Q&A, I will select questions from the Zoom chat and ask the person who submitted the question to turn on their video and microphone to ask the speakers your question live. After asking your question, please turn off your microphone once again. And please do leave the Zoom chat clear for submitting questions. All right, I think now we are ready to welcome our editors. If our editors could please join me on screen. Wonderful. So we'll get started today by having our editors give a quick introduction to themselves and their journal. We will start with Maria. Would you like to go first?
Hi, my name is Maria Polychronidou. I'm a senior editor at Molecular Systems Biology, which is an open access journal published by EMBO Press. And our journal, well, as the name indicates, publishes really everything systems biology related, including, of course, single cell biology work. And we also have a method section where we publish new methodologies with applications to systems biology. And these methodologies can be both experimental or computational or a combination of both.
Wonderful. Next, we'll have Lucia.
Hi, Shannon. Thank you very much for the invitation. So my name is Lucia Morgado Palacin, and I am scientific editor at Journal of Cellular Biology. So our journal belongs to Rockefeller University Press and is a hybrid journal. So that means that we evaluate the work in-house professional editors together with an academic editorial board. And as the name of the journal, we are interested in the cell biology works that answers fundamental questions of cell biology processes. And we also are interested in the new methods and also new data sets. We have a tools format for that. I can put the link later in the chat if any of the people in the session is interested in have a look at it.
Great, yeah, that'd be great if you can add it to the Zoom chat for everyone. One note to the audience about the Zoom chat, you will want to copy and paste any of those links because when this meeting ends, you won't have access to that Zoom chat, so do make note of that. Our next editor, Catherine, would you like to introduce yourself?
Sure, so thanks very much. I'm Catherine Brown. I'm the executive editor of Development, which is published by the Company of Biologists. So we're a small not-for-profit charitable publisher based in the UK. And so Development, again, as the name suggests, is interested in developmental biology, but we also have a strong focus on stem cell biology. And we've really sort of seen the power of single cell techniques to advance those fields over the last few years. And so we're interested in both research papers and also techniques and resources papers in the single cell field as they apply to either developmental or stem cell biology, or in fact regenerative biology as well.
Fantastic. Shiva.
Hi everyone, my name is Shiva Agrawal. I work for Cell Press for a journal called iScience. iScience is a multidisciplinary journal. We cover pretty much everything that is in interdisciplinary and on the interfaces of different fields between physical sciences, life sciences, and earth sciences. We're also open to social sciences as well as these days also medical sciences. We launched in 2018, so we're a relatively new journal from, like I said, Cell Press. And since then, we have published over 2,000 papers in these, very diverse sort of different fields within life sciences. And I'm happy to answer any questions. I will also put in the aims and scope of our journal in the chat a little bit later on. Thank you.
Wonderful. And Ines?
Hi, my name is Ines Alvarez Garcia. So I work as a senior editor at PLOS Biology. And as you all know, probably PLOS Biology is a non-profit open access journal, and we are interested in all biology. But we publish different types of papers, from research articles to methods and resources papers. So we will welcome all your papers on the exciting new methods that you have developed using single-cell biology. And I also want to mention that we are very interested in reproducibility, and we have what we call our scooping policy, in which we consider papers that have been scooped within a 6 months window. So I can encourage you to read a blog that we have called The Importance of Being Second. And I can also drop the link in the chat later.
That'd be great, thank you. And last but not least, Allison.
Hi, everyone. My name is Allison Dorr, and I'm the Chief Editor of Nature Methods. Chief Editor means that I run a team of professional editors, so we make the first and final decisions on all papers, of course, sending them to peer review as well, but we are responsible for deciding what we publish. We have just recently become, I guess you would call us a hybrid journal. Transformative journal is the terminology we want to use. So we offer subscription-based publishing, but we now also offer an open access option. And we really publish methods, software tools, comparative analyses, resources for across the gamut of basic life sciences research. And I just wanted to say, this meeting is particularly exciting for me because single-cell methods has been a major area of interest for us for a long time, not just in the transcriptomics, genomics field, but across multiple different fields.
Great, and that actually is a great segue to kick off our discussion. I'd like to ask each of you, what are the most exciting advances you've heard so far in the meeting or are looking forward to hearing about later today and tomorrow? So whoever wants to jump in first, go ahead. Allison, do you want to take the lead on that?
Sure. Yeah, I think this is a really nice meeting in that it brings together people who are both methods developers and more on the biological side. So, me personally, I'm very excited to see the methods, new computational tools being presented in talks, and really things that are, not just more practical, but are really pushing the boundaries in terms of what it's possible to learn about biology. So, some particular areas of interest for us have been spatial methods. We just highlighted spatially resolved transcriptomics as our method of the year in our January issue. Multimodal omics methods, I think, are also really interesting. Trying to squeeze as much information as possible out of, one tiny little cell. It's just so amazing what people are doing these days. But, I've also enjoyed seeing the biology talks, and it feels personally rewarding when I see, oh, this group is using a method that we published. It's actually making an impact in the community. That's great to see.
Great, yeah, and I think that's what's really exciting about this meeting is we get these technical advances that are just pretty mind-boggling to me, and then you get to see the vast amount of information and insight that can provide. What about any of the rest of our editors?
Well, I personally, so this is the first. Oh, I'm sorry, Maria.
No,
you go ahead, and then we'll go with Maria.
Yeah, all right, sorry. No, well, what I was going to say was I personally really, really enjoy the diversity in which all of these single-cell methods can be applied in so many different systems and so many different settings. And also the fact is that, systems, organs, whatever, they are not homogeneous. And this is something that, as a traditional cell biologist, this is something that we couldn't do when I was doing my PhD. And now I'm showing my age, obviously. But now the fact that you can actually look at single cells and look at how heterogeneous a population is in a sense of, what exactly single cells are doing to contribute to a certain phenotype. I find that amazing. And maybe, I'm really looking forward to see how far we can actually go for this, probably even to look at, effective therapies when things actually go wrong, just keeping in mind this heterogeneity instead of thinking, okay, everything is pretty much homogeneous in one particular place. So I find that really exciting personally.
Yeah, definitely. Maria, you had something to add.
Yeah, I mean, I agree with what was just said. I think the combination of biology and technology is very powerful, and that's a reason why I personally was very much looking forward to this meeting. in addition, of course, to the fact that single cell biology aligns well with the scope of our journal. And I think single cell biology is very powerful for analyzing both fundamental biological processes like cell differentiation, development, cell cycle, but also disease. And I think it is really nice that the meeting combines both these aspects. So I'm really looking forward to the dynamics session tomorrow, and also the computational approaches session later on, just to see what kind of new computational methods are around for interpreting and analyzing data in new ways and deriving new insights. And yeah, I cannot single out anything specific that I heard so far, but yeah, it has been a very interesting meeting.
Yeah, that's true. And that's interesting because the methods provide so much data. Then how do we manage that, and how do we make conclusions? So yeah, I think all those pieces have to come together. Lucia, did you have, sorry, Catherine, go ahead.
Well, sorry, Lucia. So I think for me, I was at this meeting 2 years ago in person in Keystone, which was really nice. And I think what for me is really great is just how fast this field is moving. And from sort of my perspective scientifically, as a sort of developmental biologist, I think the things that are really important that are really coming are both the spatiotemporal analysis that we can do now. So in terms of understanding lineage relationships at the single cell level, and also looking at not only is there lots of heterogeneity, but how is that heterogeneity organized in space and in time? And for me, I think that's sort of what's really exciting and where the field has really sort of accelerated over the last couple of years.
I completely agree with Catherine. So it's my first meeting specifically in single cell biology, and I was blown away yesterday by all the new technology that is available at the moment, and how the field has really matured from producing a huge amount of data that was very difficult to analyze to really be very clever, producing ways to look at the data in very specific ways to tackle disease and development, etc. So it's a very promising for the future, and very powerful to see what is going to come from this field. So it's very exciting.
Yeah.
I just wanted to add, for me, it's also my first single cell meeting, as in the case of Ines, and I was really amazed by how the field has combined different single cell technologies in order to provide like more clear picture of this heterogeneity that Shiva was before mentioning, and in order to infer like these cellular states that they might be like very important in the tissue architecture, and also in the, if we want like to predict how our tissue is going to behave in response to certain treatments or perturbations. So for sure, this is going to have like a lot of implications for better therapeutical strategies. And that's amazing how all these techniques are combined, and also how these advances provide like more resolution. So I'm pretty sure that we are going like to see in the next talks like very high and very exciting the advances.
That's true, and that's a really good point. Not only are we making huge leaps in the technologies, but combining all those technologies as well. And yeah, it's just really incredible the impact that can have. So our next question for discussion is, what kinds of stories then is your journal looking for at this meeting? What are you looking to publish? Do you have any special issues or anything of relevance? I know our audience probably is excited to contact you and see if they can publish their work in your journal. So what types of research are you looking to highlight in upcoming issues? Maybe Ines, do you want to go first on this one?
So we are really broad, a really broad journal, as I mentioned before, and we publish methods. So that would be one of the avenues that would allow you to publish in PLOS biology, for example. What was I going to say? Sorry, I had something in my mind.
I also wanted to add, whether you're interested more in the method advancement or the application of a method to study a disease or something
So
like
both.
that, or both?
Both. So
Okay.
we are so broad that we would be interested in both. But what I wanted to add is that because we are only an online journal, we don't have any restriction in size of papers. So you can just send us any size of paper that you want to. Sometimes actually for methods and resources papers, that helps a lot, and is one of the things that the people appreciate.
Great.
So keep that in mind.
Who would like to go next? Catherine?
I can go.
Oh, sure.
Sorry.
Shiva, go ahead.
I keep interrupting people. I apologize for this. So iScience, we're a very multidisciplinary journal, and I forgot to mention that we're also open access. And the thing is that single cell technology is literally the crossroads of all of these wonderful different disciplines. We are definitely focused both on technical papers as well as like methods papers, as well as the applications into biology. The thing is about us, I think that what makes us unique is that because we are so multidisciplinary, we are not asking for just technical papers or just biological papers. We can have papers that combine both of them. And we actually like those papers because then it kind of makes a sort of a complete story. And what we do is, in peer review, we really make sure that all aspects of your paper is looked at. So, for example, I would have a sort of like a modeling person, for example, or a technical person to look at the technical bits, and an actual biologist to look at the biology of the paper. And so we do have an all-rounded sort of peer review process that I think all interdisciplinary journals can actually already do as well. But I think that is something that is quite special that we all do. And I invite everybody out there listening, please, contact us if you have any kind of any inquiries or any questions. We are always open to pre-submission inquiries, and we'll get back to you, very quickly on that.
Fantastic. Catherine, did you want to jump in?
Oh, yeah, I can jump in. So as I've already said, we are interested in both the biology and the technical side of things as they apply, specifically within the developmental biology and stem cell fields. We do have a technique section. We do also consider computational papers that don't necessarily have experimental data in them. And actually, we run with academic editors, and we've recently appointed academic editors to cover some of these newer fields. So for computational field and for the single cell field, Sam Morris, who's going to be speaking, I think, tomorrow, joined our team recently. And so if you're sort of interested, you can either reach out to me or you can reach out to Sam to talk about your papers. And I think the other thing that I would say is that we have a pretty active review section of the journal, and that's something that I'm very involved in. And we are always happy to look at people's proposals for review articles. And so if that's something that you're interested in sort of finding out more about or in talking to me about, then you're more than welcome to get in touch about that as well. Oh, and I guess I should also say we have a couple of special issues coming up that may be of interest to people. So we're about to close submissions for an issue on imaging, and specifically looking at imaging technologies in developmental and stem cell biology. And then we've just opened a call for papers for a special issue on the influence of the immune system in development and regeneration as well.
Fantastic. Maria, would you like to jump in?
Yeah, so as I mentioned before, we also, as we heard also from other journals, we are also a very diverse journal, and we publish both studies emphasizing new biology, but also studies presenting new methodology. Our method section is also quite diverse. I will include in the chat below a single cell biology collection that we assembled with recently published papers, so that you can get an idea of the kind of studies that we publish. Yeah, they are diverse. They cover different aspects of single cell biology, from single cell omics types of studies to quantitative single cell biology, dynamics analysis in different systems. The same applies to the methods. They're also quite diverse. So we publish computational methods that are more tailored for data analysis and integration. We also publish imaging-based methods, screening platforms, anything that can have applications to systems biology. and especially for methods, because we think that it's very important to do, we try to do our best to make sure that the methods we publish can be are published in a format that makes them widely accessible and likely to be widely used. So we have also, since a couple of years, introduced a new materials and methods format for the method papers, where we allow the authors, if they wish to do that, to include their protocols in the method section, so that someone who reads their paper can really have access to all the details, all the methodological and protocol details that they need to know if they want to adopt the method in their own lab. And we are getting very good feedback regarding that format.
All right, great. Allison.
Sure, yeah. So, methods is in our name. Obviously, we're really interested in new methods, things that are novel and cutting edge, really advancing what's possible to learn in terms of biology. I think not everybody realizes that we have other formats that I think are equally important. So one I'd like to highlight is called analysis. So this would be a comparison of either methods or software tools. These tend not to be perhaps the splashiest papers, but I think they're really important for evaluating performance of methods and for, ensuring that they're reproducible, and resources as well. So we don't publish a lot of them, but again, I think, collections of tools or even, huge data sets, these are things that we are actually interested in publishing. And we also publish reviews, perspectives, and opinion pieces. And I think these are also important. They nicely supplement and sort of convey what's going on in the field. I think opinion pieces is something that we're definitely going to be focusing a bit on this year. And I just wanted to say, I feel this meeting has been a lot about the transcriptome especially, but I think what we're interested also in is the other omes that are maybe not getting as much attention at the moment. So the proteome, the metabolome, the lipidome, the glycome. I think these are much harder to study than the transcriptome, because of the diverse chemistries and the dynamic range issues and the temporal issues. But so they're still very much in their infancy, these fields, but this is something that we're looking forward to seeing develop over the next couple of years. So I'm particularly excited about that. The single cell versions of these methods.
Mm-hmm. Definitely. And Lucia, did we get you for this question yet?
Yeah, so we are a broad journal of cell biology. So as I mentioned before, we publish these papers on cell biology, but we also have this tools format for new methodologies, for new data sets, new resources, and we are also publishing the development of these new methodologies and yeah, everything with a focus on cell biology, but we have also that more methodological part in the journal. So we try to cover both the cell biology aspect, but also these new technologies that may help advance the cell biology field for analysis or for quantification. Yeah, we try to cover both. Yeah.
Great. Well, I think we're approaching our halfway point, so I think we'll transition to audience questions now. For this part, I'll ask our audience, please turn your cameras on and join us on screen. We'd love to see your faces, see who we're talking to today. You can stay muted unless I call on you to ask your question. Hello, everyone. Thanks for joining. Our first question today goes to Dagan Segal. Dagan, can you activate your microphone and ask your question?
Yeah, it's Dagan.
Dagan.
Dagan Segal. Yeah, I'm a postdoc in Dallas at UT Southwestern. And what is it that I asked? So I'm in the process right now of writing manuscript, and I'm struggling with with how to frame it, because I came up with a new analytical and an experimental pipeline, that's pretty innovative, but I also have interesting biological results. And oddly, that's paradoxical because, it seems like you need to emphasize one or the other with the, the current trend of journal formats. And I'm sure that's everyone's goal, right? Everyone comes up with an analytical pipeline with the goal of answering a biological question. So how do you see these kinds of projects, and what do you recommend? What do you think needs to be emphasized versus de-emphasized? And I see that Arjuna agreed with me in the chat that this is a major gap in journals right now.
I can answer that question quickly. So, the reason why we launched iScience at Cell Press was just exactly to capture these kind of papers. We recognize that there is definitely a gap, where there are the more traditional journals that I'm talking about, traditional in terms of around for quite a long time. They have their own sort of remit, but we are such an open journal in terms of our remit and our, the fields that we cover. I mean, what you mentioned about the fact that you've developed something, a technical sort of aspect of your work is very innovative, but also you have the biological part of it. I mean, that really sounds like something that we would like to have at iScience, because we really do want to publish, these kind of, when we talk about multidisciplinary papers, we're not talking only about papers that are in all of the different disciplines. We're also talking about the multidisciplinarity within an article. And so, yeah, so I mean, this is exactly, our aim, and this is why we exist. So.
Great. Any insights? Yeah, go ahead.
I was going to jump in. I think it is a challenge because, you do want to make a paper that's not, going to try to cover too much ground. But I'd also say that I think it's exactly the kind of thing that a lot of journals actually really want, because it's all very well developing a new technology or a new method, but unless you can demonstrate its utility, then it's not necessarily going to be as useful to the community as something that can really show a sort of biological case study where you've demonstrated the importance of that technique. So I think it is a challenge. It's also a challenge from our perspective to find the right referees for a paper in order to make sure that it gets reviewed both by people who have the technical expertise and also people who understand the underlying biology. But I do think that those kinds of papers can be some of the most valuable for the community because they both provide the technique and also show exactly how it can be useful to uncover something exciting. So I don't really have any advice about exactly how you're going to frame that. I think that largely depends on who you're most interested in reaching out to as an audience and thinking about, do you think that this is something that's a really exciting finding for the biology people, or is it that it's really the technique that is the sort of bigger advance? So it's a challenge, but I think that these are great papers that really demonstrate the utility of the technologies to a broad audience.
I would agree with Catherine completely on that. I think, we're a methods journal, but we're very excited. We're very happy when authors also choose to include novel biological results. We don't require it, but, if you want to put new results in a paper, that's great. They are more challenging to peer review. I definitely agree on that, because you need somebody who understands the biology as well as the technical aspects. And I forgot the other point I was going to make. Maybe I'll come back to that another time.
Great.
I wanted just to add, I totally agree with Catherine and also with Alison. Those are like great papers because they demonstrate how valuable is the technique. And in our case, in terms of submission, you don't need to be worried about the format because we are format neutral. And maybe after evaluation, if you submit, for example, as a tools format, we may, if that would fit better as an article, ask to the authors if they are willing to change the format, because at the end for us, what is really important is to publish this with science. So we don't really are so much worried about the type format. But I also agree with Alison that it's true that in terms of peer review, it's hard because you need to combine both expertises, and that's sometimes hard to get.
Can I ask a follow-up question?
Sorry.
Okay. Go ahead.
I just wanted to add, because I also see some comments, like Arjun made some good comments in the chat. I just wanted to say that I agree with what Alison and Catherine said. In a way, the case that you describe is like you're in a privileged situation if you can present both new methodology and new biology, because this is the more or less the dream of every journal, to get a like round complete paper that has not only new method but also a real application and new biological findings. However, the tricky thing in these cases, and actually the question that we get really a lot, is where does one draw the line in how much biology you demand the authors to present in a paper that presents new biology? And this is of course a very tricky thing to address for us editors and for the authors as well. And I think because, as my colleagues said, for those papers, we always try to get reviewers that evaluate from one side the methodology, and then other reviewers that can comment more in depth in the biology. And of course, as Arjun mentioned in the chat, there is the risk that you get many demands to extend the methodology, but also many comments to address to increase the depth of the biological analysis. So I think in these cases, it's really worth to contact the editor, explain where you think you and they should draw the line, and try to discuss with them and try to find a solution that is satisfactory. And because of course there's, I mean, I agree, there is really the tendency to try to stretch the biology as much as possible to make a strong case for the method. But usually there is an in-between solution can be found to demonstrate the potential of the method to derive new biology, but without going at length with writing a whole, I don't know, new mechanistic analysis or something like that. So I think it's very important to talk to the editors in cases like this.
I can offer you another solution in case you want to. I mean, I think that these papers are very beautiful, and they will attract a lot of audience. But you could also send us, just focus on the method, for example, at the beginning, and then we have also this format called update article, where you could actually develop the biology, and link. Both articles will be linked, like two stories, one continuing the other one. So they are updates, and they have, it's a short format, so they only have 4 figures, but sometimes they are enough to complement the method, and then you get 2 publications instead of 1.
Interesting. I do have a quick follow-up question. Sorry for hijacking the conversation, but so if you have complementary experiments that don't use your main methodology but complement your biology, so do you guys suggest de-emphasizing those if you try to focus on a methodology paper, or sort of incorporating them? It's been really hard to write. Like I read a manuscript and I keep sort of going back and forth between the arrangement of it, because
Yeah, I mean, I'm really speaking from the methods journal perspective. So I think, we probably have a slightly different perspective than a journal that has a broader scope. For us, the method has to be really well characterized. So, if it were a paper submitted to Nature Methods with this kind of situation, we would kind of err on the side of sacrificing the biology for providing more characterization of the method. It's funny. It's a nice problem to have, right? Because I feel most authors would divide this kind of thing into 2 papers, right? Try to get 2 publications out of it. But so it's nice that you do want to publish, a complete story.
Yeah.
Right. Yeah, and I think jump in really here. Go ahead, please, Catherine. Sorry, go ahead.
It's really about the audience you want to reach and thinking about. So I think in this case about really thinking about what your target journal is, but more important than the journal, who you want your readers to be. And so if you want your readers to take home the biology lesson, then those additional experiments that don't use that method are very important. If what you're expecting the sort of the main interest to come from is the method side of things, then you may want to remove those and put those into a separate paper if you feel like there's a follow-up story, or de-emphasize them in some way. So I think it's really a personal choice of who is it that you think is going to benefit most from reading your paper.
Yeah, I just wanted to just add to that as well. I think that it's really important, you know, for any journal that you would want to submit, to really look at what the aims and scope of the journal is, and every single journal has one of these. You know, just detailed out what is it that we expect. You know, if it's a methods journal, then you know exactly what is expected. If it is any other sort of journal, if you know, more of a biological slant, more of a technical slant. So if you're writing a paper, I think it's important to already have sort of a journal in mind, to think about what it is, where do you want to actually submit, and then write that paper according to that journal's style. And I think that this is, you know, like general sort of writing advice that that I think is important, because yeah, absolutely, there are journals out there that have different emphasis, and you know, it'll be good for you to know, you know, where you want to submit, so that you know what to emphasize your paper on.
Okay, great.
Thanks so much.
Yeah, and I think this discussion highlights that the editors are there for you to help shape the direction where they think it will be most impactful in their journal as well. So yeah, we encourage you to reach out to these editors for further discussion as well. Our next question goes to Mario Flores. Mario, can you activate your microphone and ask your question?
Give me a second to find my camera.
All right, great. Hello.
Hi. So probably a basic question. I have been working in single cell in the last year, and I'm getting ready to prepare to get data for a liver cancer in South Texas. And I think one of the, I would like to see a journal or an effort for what I consider is a big problem in the cell type identification, not just from the point of view of methodology, but from the concept of what is a cell. So I was wondering if there is any effort on that direction.
Yeah, I think it's a big question. I think there's also an interesting discussion to be had around to what extent it's a semantic question versus something real. I think, you know, I've been to various meetings where discussions get very bogged down in how do you define a cell type and how do you define a cell state and what's the difference between them, and, you know, and does that actually really matter? And this is where I think that really trying to link, it's very easy to take a single cell transcriptomic set of data and to say, I have X number of cell types within that, but that really becomes relevant when you can start linking that sort of molecular signature through to some sort of interesting function or lineage or whatever it might be. And so I think it's really about taking that analysis one step further and going from the here's my heterogeneity, and actually trying to understand what that heterogeneity means. And I think that, you know, people who care about this can argue all day about what do we mean by a cell type and when is a cell state a cell type. And I'm not sure that really matters. What really matters is can we ascribe functions or at least, you know, relevant differences to those different types or those different clusters.
Okay, that's a very good question, because I hear all these different nomenclature of states or subtypes or cell types. So which one is the right one to use? Like you said, probably you just gotta situate it within using the transcriptome. That is what really the data that we have in the case of single cell RNA-seq. Thank you.
Does anyone have anything else to add? All right, well, we'll move on to our next question is from Judikyle Sout. Judikyle, can you activate your microphone and ask your question? Oh, I see you're talking. Are you? Try again?
Yeah, do you hear me?
Yes, we can hear you now.
Okay. Hello, everyone. So I'm Judikyle. I'm a third-year PhD student in France. And, yeah, I don't know if my question will be precise enough, but as I said in the chat, I kind of feel like some of the single-cell omics studies are not as innovative as they were a few years ago, or at least in my case, like at the beginning of my PhD, I felt like it was a bit more, innovative. And so I was wondering if in some way the standards have been raised. Like, what do you expect from this type of papers now in terms of quality, number of replicates, and or the kind of model, this kind of stuff? Because yeah, as I go through my PhD, sometimes I doubt about this and the quality of the possibility that I have to publish these results because of this. I don't know if that's clear.
Yes, I think what are the standards for a paper? How many, as you mentioned, how many replicates, what kind of models? Are there any standards or guidelines you guys go by for what's enough for paper? What's enough innovation for paper?
So I'm not sure if you're really referring to methods papers or to results studies or kind of both. I would say, from a methods perspective, every field kind of has this trajectory where there's a lot of rapid development, a lot of innovation at the beginning, and then it kind of plateaus off and maybe there's some new methods introduced, but they're not, there's so many that exist already. They don't feel very innovative anymore. I think actually for us, I think for something that's a little bit less innovative in terms of novelty of the method, we would actually want to see higher quality of that paper, more validation of that paper, more comparison to the methods that pre-exist. I think it becomes harder to make a case for why your new method is innovative. And, again, I'm speaking from our perspective as a methods journal. For us, we care about characterization of the method. So, showing that the method works on more than one system, that it's not just kind of a specialized or one-off situation is important. But, I should let the journals that publish results studies speak from there. They'll probably have a different perspective than me.
Yeah, if I can add to that. So I totally agree with Alison on what she said about methods. And with biology, I think it's a bit the same. I guess when the first single cell transcriptomic studies, for example, started coming out, it was sufficient to describe some new population. So everything was very novel. So the novelty in itself was a very important factor to allow publishing this kind of papers. I think now the standards are becoming higher. So in the same way that Alison described for the methods, I think a stronger case needs to be made for a data set that is generated in a given system, or if this particular system has relevance for disease. Sometimes like cell lines are not sufficient. It's more appropriate to go to either organoids or maybe disease model, or even patient data is quite common these days. I think it's very topic specific, and yeah, I'm not quite sure exactly in which particular biological context you are referring to, but yeah, and I don't know how much I answered your question.
No, that's a good answer. I was thinking about transcriptomics in particular, as Alison mentioned earlier. Many of the talks of yesterday and tomorrow have been about transcriptomics in particular. So I was thinking about this field in particular. And a few years ago, there were some studies that have data, that were published in very good papers and that were reference studies, kind of. And so nowadays, we can quite easily produce such a data, but this would not be sufficient to publish in such good papers. And so there is this shift, and not this shift, but this natural evolution of the overall value of your research. And I was just wondering about all of this But yeah,
I think it's exactly what you say with the natural evolution of the field, and perhaps in some cases, for example, in the beginning, it was sufficient to describe the existence of certain populations and subpopulations, but now there's more demand in knowing, like, for example, what is the function of these populations, what are they doing, why are they different, and things like this.
I'd also just add that I think it's very hard to generalize, because I think it depends a lot on the system in which you're working. So if you've got easy access to tens of thousands of cells that you can purify and separate easily, then it becomes less hard to justify doing your single cell analysis on a large population or doing multiple replicates. If you're working with a very rare cell type, or with human primary samples, or whatever it might be, where it's much more challenging to get hold of the material, then I think that editors, journals, reviewers are hopefully understanding of that. So I think it's really hard to generalize, because it's really going to depend on your system and the availability of material, and how difficult that is to come by as well.
Okay. Well, indeed, that's what in my situation, I think I have some cool features about my experimental design and all of this. I am working with primary tissues, and so I think it's a cool thing, but also it has been difficult, and the technique is expensive, and I don't have that much money, but that's like everyone. So I have been naturally wondering this kind of thing as my research goes on. But I'm kind of reassured also, because you're confirming that, of course, this is very specific to the study, and there are some specific features about all kind of project and research, and I'm sure I will be able to publish it anyway, of course. But I was generally wondering about all of this.
Yeah, and I think that's a good point. We do see a lot of methods development, but now we're seeing the application of those tools to primary tissues, which is really exciting and adding so much insight versus just studying cells in the lab. So I think that will reveal a lot more to us as well. Okay, our next question goes to Michael Casey. Michael Casey, can you activate your microphone and ask your question?
Hello, yes, can you hear me?
Yes.
Hi, I think this is probably mostly covered by the first question, but I'm a computational methods developer for single-cell transcriptomics, and I was a biologist, but I'm now technically a PhD mathematician, and I've noticed there's a lot more maths in the field, but it's often in the supplementary information. And often I feel like the maths is what's been the real result of the paper. Like whenever I see a new method, I almost skip directly to the supplement, because that's what I see as the real paper almost, which is a bit of a weird situation to have a paper and then a supplement that you're needing to skip to actually understand the paper. So how do we go about actually integrating this growth of biomathematics into biology? As biology is becoming mathematical, and our current setup doesn't seem quite equipped to deal with that. I mean, obviously because it's new, but Sorry, that was a splutter of words. I'm sure there's hopefully a question in there.
I can take this one. Again, speaking from my journal's perspective, and this is not unique to math. I mean, we publish methods that are heavily rooted in chemistry or physics, and those communities also complain. Why are all the important details being put in the supplement or the methods section and not highlighted in the main paper? I think it's what you want to highlight. We're a methods journal for biologists. So what we want the main paper to show is, the validation of this method, and in a sort of way that biologists will understand, a figure or an image that shows, shows the kind of results that you get using this method. So, unfortunately for us, that's kind of the reality. A lot of the math itself will be kind of in the methods section or the supplement. But we hear you, and we're willing to work with authors if they think it's really important to show some of the math in the paper. It's a process. We do work with authors depending on what they want to highlight. but typically it's, that stuff unfortunately does get put in the supplement. But, if that's what you think is really important to highlight, I think, then it's a matter of choosing the right journal and just knowing what the journal is focusing on and what they want in that main part of the paper.
I think it also depends of your skill. So if you want to obviously reach a very broad audience and you want them to understand your maths or your computational method, you'll have to be very skilled if you want to put it in the main body of the paper to make everybody understand and to reach. I mean, we have a very broad audience, so we have exactly the same problem with, math and computational biology is a bit more extreme because there are not many people that understand if they're not really into the topic, but a specialist, very specialist field, they all also struggle to make everybody understand what the science is about in the paper that they have published. So my advice would be to be a good writer and be able to show everybody what you want to show. Like, try to think about, just ask advice to other people in other fields for the writing of your paper, and see if they can understand. I mean, I can tell you the example of try to tell the story to your grandma, but I think in this it's a bit of extreme to make your grandma understand computational biology. But it's a clear case of how you want to reach your audience. You have to be broad and very well.
I think the other thing. Sorry, Inez.
No,
part of the problem is on our side as well, from the journal's perspective, is that, possibly slightly outdatedly, we still call it and refer to it and think of it as the main paper and the supplemental data, and that the supplemental data is sort of less important. And the benefit you have is that all of that stuff now is equally accessible. So, maybe part of the challenge is for us as publishers as well to think about how do we provide that information in the most accessible way without making it feel like it's totally relegated to something that, you don't need to bother to read this because it's only supplemental. And, we struggle with this with all sorts of things with, until recently, all of our movies were called supplemental movies, even though, for developmental biology, the imaging is super important. And so, I think it's a process of thinking about how can we better present that? And also how can we make all of the underlying code, all of the underlying maths most easily available? And, we still fall into the trap of providing some of this as a flat PDF, which is not hugely helpful. And so I think there's also an onus on us to think about how do we better present that so that it's as accessible to sort of as broad a group of people as it can be.
I have to add also that, I think the writing, this is what Inez mentioned. I definitely agree with that. For a multidisciplinary journal like iScience, I think that it's important to bring the concept of what you've done to, multiple different audiences that are also multidisciplinary, just like you are. And there's there is a skill to that that is, that should be cultivated, especially if you're on the interface of two different fields. It's not easy, but it can be done. And I think that a lot of the papers, if you look at some of our articles, you'll see that, a lot of people have navigated that fairly successfully.
Yeah, that's a really good point. And I think it brings up a common theme we've heard in a lot of these answers is publishing and writing your work isn't just about telling your story. It's about who you're telling your story to and who you want to tell it to, and framing that discussion. So yeah, it's definitely a challenge in a lot of cases, but I think that's another thing that editors can help you with insight into as well.
Absolutely, yes.
Great. We have another question from D Who. D Who, can you activate your microphone and ask your question?
Yeah, I guess I had two. Okay, I'll just ask the protocols one first. Does having a protocols.io protocol help for like methods, papers, or sections?
Yeah, I can answer that one. Yeah. Sorry. So we at Molecular Systems Biology and EMBO Press in general, we have partnered recently, well, actually it's not even that recently anymore, a couple of years ago with protocols.io. So we encourage authors to include links to protocols.io to their method section. And as I also mentioned in the introduction, we also allow the authors to include the protocols directly in the method section, which I think, as I said in the beginning, it facilitates adoption of the method by other labs, but in a way, it's also much easier for the authors if you want, because usually I think what they have at hand is a detailed protocol. And then when we ask them to write the methods, we ask them to artificially condense that very structured protocol in a narrative that is a bit artificial for describing methods. So yeah, it has been adopted quite a lot by authors who publish with us. But we also at the same time allow and even encourage protocols.io. And I think it's very helpful, especially for methods, because it makes all the sense to have details and protocols published in your paper.
I absolutely agree. So we also encourage, we have a partnership with protocols.org and all PLOS journals, basically. So we always encourage you to submit your protocol there. And it works really well.
Okay.
Yeah, I would agree with that. We have a product at Springer Nature to Protocols Exchange. We don't require that people use that one. They're free to post their protocol anywhere they like. You know, it doesn't have to be our own repository. But I think it's really important. I mean, we've been asking experimental methods papers. We've been asking the authors for many years to write up protocols and submit it as a supplementary protocol. So it's great now that the community has all these resources. There's kind of no excuse for not doing it anymore. I think it's really important for other people being able to increase the impact of your method, having other people actually be able to use your method.
And then it says, like, if it works for them, it'll say, like, works for me. And then, yeah, the more it works for, the better. Yeah.
Definitely.
But at CellPress, we have a journal called Star Protocols, which does fairly similar things. So you check that out. I'll put the link in the chat.
Great.
Yep, I saw that for, mosquito miniaturization. Yeah, I guess. And the second thing kind of briefly is Allison mentioned the interest in other omics like lipidomics, metabolomics, proteomics. What about epigenomics?
Yeah, I probably missed that one.
Okay, yeah.
I'm a chemist by training, like a protein chemist by training. So I kind of always tend to go that direction. But yes, of course. Okay. All the omics.
Okay, that's good. Thank you.
Great. Any other editors want to add to that?
I think Alison covered it all very well.
Okay, perfect. All right, well, we are approaching the end of our time today, and we have no more further questions currently from the audience. So we'll wrap up, but I would like to highlight that Arjun mentioned also in the chat that there's been a Slack channel set up to continue the discussion. So if you think of any other questions, we'd love for you to join us there and continue this conversation with the editors, and we welcome the editors to check that out as well. I think you all should have received an email about it about half an hour ago or so as well, but the link's also in the chat. We also have the one-on-one connect feature where you can book private appointments with some of the editors at the meeting. So we encourage you to check that out. And our networking session is tomorrow during the break between sessions. It's on the agenda page. And in that you can have small group conversations and kind of mingle with other colleagues and participants. So we really encourage you to check that out as well. So finally, I'd love for us all to activate our microphones and give a round of applause to our editors for all these wonderful insights and their time today. Thank you, editors. All right, and enjoy the rest of the meeting. Bye, everyone.
Thank you.
Bye.
Thanks, bye.